Swayam Prabha. Digital India. Educated India.
Hi everybody, this is Dr. Ravindra Maradi, Associate Professor in the Department of Biochemistry, Kasturba Medical College, Manipal. In this hour, I will be discussing about disorders of ammonia metabolism. Before going into the disorders of ammonia metabolism, we have to just look at how ammonia is metabolized. Major chunk of the ammonia is metabolized and excreted in the form of urea in the urea cycle. Around 86 to 90% of ammonia is excreted in the form of urea, and this is the major route of excretion of ammonia. Any defect in the enzymes of this urea cycle leads to the disorders, leads to increased accumulation of ammonia in the body and hyperammonemia. So, first we will see how this is the normal metabolism of ammonia, how ammonia is converted into the urea. This ammonia metabolism, the excretion of ammonia, formation of urea takes place in the liver, and the reactions of this conversion of ammonia into urea takes place both in the mitochondria as well as cytosol. So, what happens in the first reaction? The ammonia that is produced in the body, that in different tissues, it comes back to the liver. It may be from the skeletal muscle, it comes to the liver in the form of alanine, or it may be from kidneys, it may be from intestine in the form of free ammonia, or most of the tissues they give their ammonia to the liver in the form of glutamine. That is the major transport form of the ammonia. They all come to the liver, and one of the free ammonia that is donated is from the action of glutaminase and from the glutamine, from the both in the kidney and intestine, or glutamate dehydrogenase enzyme acts on the glutamate in the liver to produce free ammonia. So, this ammonia is one of the source of nitrogen of the urea. Urea has got two nitrogens, two amide groups, and one of the amide group comes from this free ammonia that combines with the carbon dioxide in the presence of ATP to form carbamyl phosphate, and ATP is converted into ADP and PI. The enzyme required for this action is carbamyl phosphate synthetase 1. So, when we say carbamyl phosphate synthetase 1, there is another enzyme carbamyl phosphate synthetase 2 that is present in the pyrimidine metabolism, and this enzyme is activated by N-acetyl glutamate. N-acetyl glutamate is the obligate activator of this carbamyl phosphate synthetase 1. What we mean by obligate activator? Obligate activator means without the presence of this enzyme will not work. Like the key of your car, without key the car will not start. Like that, without this N-acetyl glutamate, this enzyme will not work and ammonia is not converted into carbamyl phosphate. In the next reaction, carbamyl phosphate combines with ornithine to form citrulline in the presence of the enzyme ornithine transcarbamylase. This enzyme converts carbamyl phosphate plus ornithine to form citrulline, and this citrulline comes out of the mitochondria into the cytosol. In the cytosol, it combines with the aspartate. Aspartate is the donor of second nitrogen of urea. The first donor of nitrogen is the free ammonia, that is from glutamine or glutamate by the action of glutamate dehydrogenase, and or glutaminase, that is from the glutamine, and the second nitrogen comes from this aspartate. This aspartate combines with citrulline to form argininosuccinate and requires a ATP, and the enzyme required is argininosuccinate synthetase. Then argininosuccinate acted upon by argininosuccinate lyase enzyme to form arginine and fumarate, and this arginine is hydrolyzed by the enzyme arginase to form all important molecule urea and resynthesis of ornithine, and this ornithine goes back into the mitochondria and the cycle will continue. So, ornithine is a byproduct that is produced and it is reutilized again, like the oxaloacetate that is produced in the TCA cycle, that is how it is reutilized. Similarly, ornithine is utilized and the urea produced is dumped into the blood and taken up by the kidney and excreted in the urine. And this fumarate can be converted back into malate, malate will converted back into oxaloacetate, and then by the transamination reaction oxaloacetate is converted into aspartate, again it can be reutilized. This is the normal conversion of this ammonia into urea, that is urea cycle. Any defect in one of the enzymes, it causes disorders related to this urea cycle. We will see one by one. Before that, why this disorder? As I already mentioned, there may be a genetic deficiency of any one of the enzymes of the urea cycle that causes this disorders, and most common one is the ornithine transcarbamylase deficiency. That is the second enzyme of urea cycle is the most common deficiency seen in this disorders. And what happens if there is enzyme deficiency? What happens? The problem that is happening is the ammonia is not converted into urea. Especially the first two enzymes are very important, because it increases the blood ammonia level, and as you all know that ammonia is very toxic, it increases the body pH and crosses the blood brain barrier, and it causes lot of problem like cerebral edema, convulsions, coma, death can happen with this. So, usually this genetic disorders deficiencies occur early in the life of the newborn, and that causes increase in the ammonia level in the blood, that is hyperammonemia. So, in the first week of few weeks of life, these signs and symptoms will be observed, and the babies will suffer from vomiting and cannot eat properly, failure to thrive, and protein intolerance, because if you take more protein rich diet, there will be more amino acid load, more amino acid catabolism, more load of ammonia to be detoxified, and it causes the intermittent ataxia, irritability, lethargy, mental retardation, and if not treated, coma and death. That is why these disorders are very important to diagnose early in the life and start treatment, because you can prevent this all the mental retardation and other things. So, what are the disorders? The first disorder that can happen, if you have seen that normal metabolism of this ammonia. So, you just put a block in this reaction. First reaction is ammonia is converted into carbamyl phosphate by combining with carbon dioxide and ATP by the enzyme carbamyl phosphate synthetase 1. If you just put a block in this reaction, what happens? This ammonia is not converted into carbamyl phosphate. The ammonia level will increase, and if the ammonia level will increase and it backs up, glutamine is also level increases in the blood. And if you look at the blood picture, urea is not synthesized and blood urea nitrogen level decreases, and this is called hyperammonemia type 1. As the name indicates, the disorder name, hyper means increased, emia means in the blood, ammonia. Increased ammonia level in the blood is caused due to the deficiency of the first enzyme of urea cycle, that is carbamyl phosphate synthetase 1. So, this disorder is called hyperammonemia type 1. Next is second reaction is carbamyl phosphate combines with ornithine to form citrulline by the enzyme ornithine transcarbamylase. If you just put a block here, what happens? Carbamyl phosphate will not be converted into citrulline and increases the level of carbamyl phosphate, and also accumulation of this leads to increased ammonia, increased glutamine, and decrease in the production of the blood urea nitrogen. So, this disorder is called hyperammonemia type 1. It is similar to type 1, but this type 2 it differs in this is there is accumulation of this carbamyl phosphate that spills over and comes into the cytosol, and this enters into another cycle, another pathway that is going on in the cytosol, that is pyrimidine metabolism, and that it increases the level of orotic acid. So, when you can how you can differentiate between type 1 and type 2 is the presence of orotic acid in the increased orotic acid in the blood or in the urine, orotic aciduria, that differentiates between hyperammonemia type 1 and type 2. In type 2 deficiency, you see orotic aciduria, but not in type 1 deficiency. Next, the enzymes that are present in the cytosol, citrulline combines with aspartate, which is the donor of the second nitrogen, and citrulline combines with aspartate to form argininosuccinate, and the enzyme required is argininosuccinate synthetase. It requires an ATP molecule, and if you put a block in this reaction, citrulline starts accumulating. It will not convert into argininosuccinate, and this disorder is called increased citrulline, is called citrullinemia. Okay. This citrullinemia is the third disorder of this ammonia metabolism. Then the next is argininosuccinate is lyased by converted into fumarate and arginine by the enzyme argininosuccinate lyase, and here the aspartate after donating the nitrogen is converted into fumarate, and you get the arginine here. And if you put a block in this, argininosuccinate is not converted into arginine. So, this disorder is called argininosuccinic aciduria, acidemia leading to aciduria. Argininosuccinate level increases in blood and then spills over into the urine. Okay, this is the fourth disorder of ammonia metabolism. And the last one is arginine is acted upon by arginase. Hydrolysis of this arginine takes place to form urea and ornithine. And if you put a block here, what happens? Arginine level increases in the blood, and this is called argininemia. The first two reactions that are taking place in the mitochondria are very important. They are more severe in nature compared to the downstream enzyme deficiency, because some amount of citrulline formed that contains some nitrogen that can be excreted in the body. So, hyperammonemia type 1 and type 2 are very important. As I told already, hyperammonemia type 2 is very common. So, we will see in little bit in detail. And ornithine transcarbamylase enzyme deficiency causes hyperammonemia type 2. Symptoms is increase in ammonia level in the blood, it causes increase in glutamine level because urea is not formed, blood urea nitrogen decreases, and there is orotic aciduria because carbamyl phosphate that is accumulated spills over into cytosol to be used in another pathway that is taking place in the cytosol is pyrimidine metabolism, and it causes cerebral edema, lethargy, convulsions, coma and death. So, what is the reason to explain? Already I told, ammonia carbon dioxide combining forms carbamyl phosphate by carbamyl phosphate synthetase 1, then it goes into the urea cycle, citrulline is formed by ornithine transcarbamylase and next reactions forms urea. So, if there is a problem in this second reaction, okay, if you put a X mark here, what happens? Carbamyl phosphate is not converted into citrulline and increase in carbamyl phosphate level spills over into pyrimidine synthesis and increases orotic acid synthesis, and this is the distinguished feature of hyperammonemia type 1 and type 2. If you see orotic aciduria, this is hyperammonemia type 2. Without orotic aciduria, that is hyperammonemia type 1. To summarize, so we have seen 5 disorders. First 2 are in the mitochondria and next 3 are in the cytosol. Among them most common is type 2 disorder, and these are the defective enzymes in what is the main substrate that is accumulated. And here in hyperammonemia type 1, the defect is carbamyl phosphate synthetase 1, and then the accumulated substrates are ammonia and glutamine. In type 2, ornithine transcarbamylase deficiency, ammonia is increased, glutamine is increased, carbamyl phosphate level increases, and that spills into pyrimidine synthetic pathway to produce increased orotic acid also. In citrullinemia, the defective enzyme is arginosuccinate synthetase, citrulline level increases and it can get excreted into urine. Arginosuccinic aciduria, the enzyme deficiency is argininosuccinate lyase and the substrate accumulated is argininosuccinate, and the last disorder is argininemia acted upon by arginase and the accumulated substrate is arginine. I hope this lecture will help you to understand the disorders associated with ammonia metabolism. Thank you.